I'd like to introduce my wonderful friend and colleague, Rob Malenka. He's another one of these doctors. Highly celebrated scientific career in National Academy of Sciences. He's the Nancy Friend Pritzker Professor in the Department of Psychiatry and Behavioral Sciences, a leader of the Stanford Neuroscience Institute, and I think his dinners for years actually led to the initiation of the Stanford Neuroscience Institute, an associate chair in the department, and he's really focused on fundamental mechanisms of disease and health, brain function, and I want to welcome him to the stage.
Ooh, my time has gone down. This is it. So we're behind schedule, so I will try to limit my comments to 15 minutes. It used to be 17 minutes, but now it's 15 minutes, which is fine. So I'm going to change the topic just a little bit. I think I'm the first, other than Ed Boyden, the first what I would call basic neuroscientist who focuses my work on animal models and animal work, primarily in rodents. And my hope is to help you understand why this kind of fundamental, foundational neuroscience is really critical for advancing the types of topics you've heard about from all of my clinical colleagues. You've seen, this has already been covered by several different speakers. So all it points out is, once again, brain disorders affect an enormous segment of our population, not only in the United States and in the world. I think the one, and if you look at that number, we are grossly underinvested in trying to figure out how these diseases are caused and how to treat those diseases. The other point I'll make is think about having a disorder of your brain. You know, we don't want to be competitive with our colleagues who study very important diseases like cancer or infectious disease or metabolic disease. But when all is said and done, I mean, if your brain is not functioning normally, you are not yourself. I can't imagine being so depressed that you commit suicide. Think about that experience. I can't imagine having a disorder like severe forms of drug addiction or OCD, where you literally have no control over your behavior. You cannot control it anymore. And again, working on cancer is obviously incredibly important, and solving HIV, obviously incredibly important. But if I had Sophie's choice, the last thing I would want is my brain not functioning normally, because you lose yourself. And that's why we are all so passionate about this topic. Okay, the challenges are pretty obvious. The brain, especially the mammalian brain, is the most complicated entity in the universe. It is overwhelmingly complex. It is mind-boggling complex. The diagnoses we use, especially in psychiatry, are often ambiguous because we use these checklists. Let's pick on depression. Two people can have depression and have almost no overlap in their symptoms. And that's why, as we've heard from Lee Williams, as we've heard from Amit Etkin, as we've heard from Tom Insel, why it is so important to begin to develop tools that allow us for better stratification of patients and more precise diagnosis, the precision psychiatry that Lee just talked about. Another problem is comorbidities are common, meaning it's very uncommon to only have depression or only have addiction or only have anxiety. And then really what we do primarily in psychiatry is we treat symptoms. We talk about diseases, but we actually treat symptoms. And the symptoms cross diagnostic boundaries. So I picked on a symptom called anhedonia, which is the inability to experience pleasure or reward or joy. And that's a cardinal feature of depression, but it happens to Parkinson's patients. People with schizophrenia suffer from anhedonia. So even the symptoms we're trying to understand the pathophysiology of and treat do not segregate with our diagnostic nosology. And finally, A different point I'd like to make is when I was a medical student here at Stanford a long time ago, trying to decide whether to go into neurology, neurosurgery, anesthesiology, ophthalmology, I chose psychiatry, one, because I was fascinated by the topic, but of equal importance for me is by studying individuals and animals with mental illnesses, it allows us to also study what makes us human, it allows us to study the neural mechanisms that allow us to be happy or sad. It allows us to try to understand the neural mechanisms that mediate empathy and compassion and aggression and anger. And with all due respect to my non-psychiatric colleagues, if you're a neurologist, you don't get to study what I think are the most fascinating aspects of human behavior. If you're a neurosurgeon, unless you're really unusual, you don't get to study those, and et cetera, et cetera. Okay, so treatments. We obviously, like every branch of medicine, we need new treatments, and the available treatments have lots of room for improvement. I do want to defend the field and say our treatments actually aren't bad. And if you actually really look at them and compare the efficacy of the treatments we can give our patients with mental illnesses, they actually compare reasonably favorably to a lot of the treatments people use for treating diabetes or respiratory illnesses or metabolic disorders. But as we've heard over and over again, there's a lot of room for improvement. I'm going to be focusing a little bit on a topic we haven't heard that much about, is drug development and drug discovery, and I'll explain why in a second. So if you think about new medications for the treatment of brain disorders in general, in particular mental illnesses, it's really a horrible history. There's basically been a major, I say it's been stalled. Many of the big pharma have deserted putting any resources into efforts to make new drugs for the brain. They've either stopped their internal neuroscience efforts, they've looked for external collaborations, and if anybody's interested, and some of you may know quite a lot about this, I'm happy to talk about my view of why that has happened, because of the corporate structures of pharma and because of the challenge. And in fact, if you look at the history of psychopharmacology and the treatment of the development of drugs for the treatment of at least mental illnesses, I can only think in the last 70 years of one single drug that was really developed based on understanding of pathophysiology and with a true scientific basis. The history is astute clinical observation, but accidental clinical observation. That's how lithium was discovered for the treatment of bipolar disorder. Then trying to understand that drug's mechanism of action, which was just studied serendipitously. The latest example of this is ketamine, which there's almost a hysteria about how ketamine may be a rapid treatment for depression, perhaps OCD, and that's wonderful. We need to study it, but it was discovered accidentally. We have no idea how it really works. So the history of pharmacology is find a drug by accident, spend a lot of time trying to figure out how that drug, in quotes, works, which means identify the molecular target. We really actually have no understanding of how any of our drugs really work in the true therapeutic sense. And then, if you're a pharmaceutical company, make Me Too drugs that will become blockbusters and make you billions of dollars, and try to separate those. I'm a believer, as I think Ed Boyden is that the breakthroughs in neuroscience methods offer an opportunity in this space. They're going to open the doors to new approaches for many different types of treatments. I've listed some of the new methodologies on this slide. Ed talked about some of them, optogenetics, something called chemogenetics, et cetera, et cetera. Tom Insel very eloquently described how the use of mobile digital apps and machine learning, Lee talked about this a little bit, also may be able to really advance and revolutionize our ability to get our currently available treatments out there and get bigger market penetrance. And I think that's hugely important because, as I said, the treatments we have now aren't bad, but they're not getting to a lot of the people they need to. And the type of work Tom Insel's working on and others, telemedicine, might really solve that or might help with that. But for advancing new treatments, or developing new treatments, as you've heard from many of my previous colleagues, Ed Boyden, Amit Etkin, Lee a little bit, Ruth a little bit, we all think that understanding this, quote, circuit pathophysiology mediating prominent symptoms is the way we have to go. And the way we use the term circuits may be a little bit confusing. It's confusing to me, and I'm a card-carrying neuroscientist, because a circuit can be 3 cells in one part of the brain communicating with 3 cells in another part of the brain. That's a circuit. Or it can be 17 million neurons in 27 different brain regions all communicating with each other simultaneously. But nevertheless, that's what we're dealing with. Trying to understand this complex communication between different brain regions and how that might work. So, whoops, did I miss a slide? So Ed already described the really important development and discovery of optogenetics, which happened here at Stanford, led by Carl Deisroth, working with Ed Boyden and Feng Zhang. And I just highlight optogenetics because it's really a tool now that, in terms of the basic animal neuroscience work, has revolutionized what we can do. And I'll give you an example of that. So as Ed pointed out, this just involves expressing in light-sensitive ion channels or pumps in neurons using delivery systems such as viruses. And these proteins are completely inert until you shine the appropriate wavelength light on them, and then they allow you to control the activity of individual nerve cells in an awake behaving animal or brain. And we have many very sophisticated ways of using these tools now in very precise experimental manipulations. And you can do this, as I said, in awake behaving mice. God, my time is going by fast. I'm going to use drug discovery as an exemplar. This slide is just, I'm used to giving this talk in front of medical audiences, and it's required that you disclose possible conflicts. It's also to convince some of you that have invested or worked in this space of drug discovery, biotech, that I am not a naive academic who has no idea about the overwhelming challenges pharmaceutical companies, biotech companies face in trying to develop a drug that will actually be successful in a phase 3 trial and approved by the FDA. So I've been a high-level advisor for many pharma, for various small biotechs. I started a biotech myself called Circuit Therapeutics with some Stanford colleagues 6 years ago, and I'm working on starting another biotech with Carl Deisroth and some other colleagues actually right now as we work. So my goal, as I said right now, is to convince you why investing, whether it's philanthropically, whether it's through grants, whether it's through commercial entities, in foundational basic neuroscience. And I'm going to try to convince you that what we need to do as a field is integrate this foundational basic, primarily animal neuroscience, in a much more effective way with the human neuroscience efforts that you've heard about throughout this morning. And that is a very important ecosystem or platform for advancing what we all want to advance. That is improving diagnosis and treatment of mental illness, understanding resilience and wellness in a much more sophisticated way. So one way, and I'm rushing through this because the time's going by fast, one way to do this is to use these modern tools of neuroscience, optogenetics, and other ways of tracing circuits in a rodent brain, and perhaps in the non-human primate brain, in a fairly sophisticated way. Figure out the role of that specific circuit and do a behavioral analysis of that, and then use that information and collaborate with our colleagues who are working in human beings, who are doing the type of work that Lee Williams talked about, that Amit Etkin talked about, to help them advance the types of imaging, the types of circuits, the types of manipulations they're going to do. So here's just one example from my own lab where we were working on a set of dopamine neurons. Most of you have heard of the neuromodulator dopamine. We were interested in the question of what are the neural circuits that make a mouse happy or sad, make a mouse feel good or make a mouse feel bad. And what we were able to do is use a virus tracing technique to identify all the brain areas that send inputs into these dopamine neurons. We were then able to select some of these inputs and in a very precise way use optogenetics to activate these different sets of specific inputs. And what we found is that one set of inputs made the mouse happy, it was reinforcing. A different set of inputs made the mouse unhappy, it was aversive. We were then, with a lot of other work, it allowed us to begin to make what I would call a map of novel circuitry that had not been elucidated before. We heard a talk, some of us last night, about cartography and making new maps. This is a map of a novel circuit in the mouse brain that is probably the equivalent of a 16th century map of the world. It's still primitive. We need to know a lot more. But nevertheless, it's novel, it's new, and so one could argue, who cares? Well, luckily, I had a colleague who was then in psychology. Am I really out of time already? Who took this information and studied some of these circuits in Stanford undergrads. And in particular, he studied a circuit that we discovered that makes the mouse feel bad. So the question is, in a human brain, is the same thing going on? So he took some Stanford students, he gave them modest electric shocks, which was a modestly aversive experience. And what this tells you is that he basically saw some of the same functional connectivity in this human brain that we had discovered in the mouse brain. And I would argue it's this type of iterative, starting with doing manipulations in a more simple system like a mouse, and then seeing if that information can be applied to the human. So I am actually way out of time. I thought I had 20 minutes, not 15. I'll try to wrap up in about 2 minutes. I also believe that this kind of approach can be used for new platforms for identifying novel targets and making drugs to those targets. And the workflow is kind of shown here. You can define a circuit that's mediating a specific symptom. You can then go into the key cell types within that circuit, defined in a much more sophisticated way than we were ever able to do before. And in the next slide, and then I'll wrap up, you can do gene expression profiling in that specific cell type, knowing its detailed connectivity, knowing that when you activate it or inhibit it, it has a certain behavioral role. Then based on that target you have selected, you can validate that target in a variety of different ways, using genetic manipulations or using what we call as tool compounds. And this slide just describes to you how you can begin to do gene expression profiling, both in the rodent brain and monkey brain, and there are companies now being started and academic labs who are trying to do something similar with human brain tissue. Obviously, with human brain tissue, it has to be taken out of the individual, so this is post-mortem tissue. But again, the goal is by integrating the data that is collected from rodent models where you can do slightly more sophisticated kinds of analyses and then see how that crosses into the human brain, it's going to accelerate the ability to identify novel targets, make new drugs. There are new methods coming on. I'm going to move on from this. Sorry, guys, those of you who wanted to hear about my MDMA work, I met some of you in the bar last night, but I don't have time. I also think we can learn a lot from studying drugs that already exist that we know have very powerful effects on the brain. And if we can understand how those drugs work, we can make better versions of those drugs that may not have abuse liability and may actually be therapeutically beneficial. This just says you can also use these approaches, what I call circuit busting approaches, to define. This has worked from an ex-postdoc of mine who's the director of the intramural program at the National Institute on Drug Abuse, Antonello Bonci. And what Anto did is he used the kinds of tools that Ed Boyden talked about, I tried to describe to you, to define novel circuit connectivity in a mouse addiction model. And then based on that, he predicted that if he collaborated with clinical colleagues, maybe they could use transmagnetic stimulation to repair this circuit pathology. And it's really early days, but what he found in a study in Italy is that by targeting this brain region and this connectivity that he had elucidated in a mouse model, he was actually able to use TMS to reduce the relapse in a number of cocaine addicts. Really early days, but again, another example of how I believe you can go from fundamental neuroscience and neurocircuit elucidation, even in organisms as simple as a mouse, and build up that knowledge base and apply it to human beings. And you can also, you're going to hear from another colleague tomorrow, I think Casey Halpern, who's using this kind of approach to improve deep brain stimulation kinds of approaches in neurosurgical patients. So this is my last slide, and this actually is ripped off from Lee Williams. Where's Lee? So the idea that we all believe in is that by understanding more and more about the circuit pathology mediating prominent symptoms of mental illness, we will eventually be able to have more precise diagnoses that will lead to more precise treatments. But I personally think the bottom sentence here, this is only going to happen if we continue to make long-term investments in fundamental neuroscience. So I apologize for going way over. Give me the hook. All right. Yep.